morning. Science. Science is the exploration, the investigation of the natural world. The natural world is what inspires us. It's a tangible world that stimulates all of our senses. It's a world where we're allowed to ask questions, be curious and inquisitive about everything around us. But science is changing. Biology, in particular, is changing. The natural world that we are all familiar with is not as obvious as it might seem anymore. Biology is among the youngest of the natural sciences. It's much younger than chemistry or physics. Biology has its roots back in 1753. A man named Carolus Linnaeus was the first man to, and this sounds revolutionary, he was the first man to say that one organism can be classified with another organism. In 1859, Charles Darwin gave us the theory of evolution by means of natural selection, a theory that was founded in his meticulous observations of the natural world. And in 1865, a monk by the name of Gregor Mendel, unbeknownst to him, became the father of modern genetics, again, with observations in the natural world. All three of these men have that one thing in common, is that they were observing that tangible world around them. They were manipulating the things that they worked with, and they were allowed to make observations and allow their curiosity to wonder and ask why and how. That all changed 200 years after Linnaeus. In 1953, when an American and a Brit named James Watson and Francis Crick proposed the double helical structure of DNA. That was 1953 that we discovered the double helical structure of DNA. We knew more about the inner workings of the atom before we knew about DNA. This was the game changer for biology. The natural world was no longer limited to the big things, the animals, the plants. It was now in the realm of the molecule. Where have we come? It took us 200 years to go from Linnaeus to Watson and Crick. In the last 60 years or so since Watson and Crick, we have come a long way. In 1962, we coined the term molecular biology. In 1982, we have genetically modified organisms. The idea that we can take a gene from one organism and put it into another. This is a topic we still see in the headlines today. In 1984, DNA fingerprinting, where we can take a little molecule from any organism and compare it to another organism. And in 1990, gene therapy, the idea that we can correct genetic material that has errors in it. In 1996, Dolly the sheep, the very first cloned organism, where we took a cell from an adult and turned that into another copy of that same individual. And finally, in 2003, the greatest project in biology as we have ever accomplished thus far is the Human Genome Project, the decoding of the very genetic code that defines us as human beings. The challenge with this is, as a teacher, how do we make these discoveries and this world tangible? If students are going to truly learn biology, we need to make this molecular world and this atomic world tangible. Now, I'm seeing some people might be kind of confused. How small are we really talking here? What is a molecule? Well, journey with me here. This is the tangible world. We have a coffee bean, a grain of rice. We are going to zoom in to the realm of the molecule. Our first stop here is amoeba and paramecium, those little beastie boys we see swimming around in every biology classroom. We then zoom into the human cells, skin cells, red blood cells, egg and sperm. And now we have the bacteria, the smallest of all living cells. Just now, we are going to start seeing the realm of the viruses, those little pathogens we love to hate. And now, and only now, do we start to see the first molecules, the ribosomes, the proteins, nucleic acids, carbohydrates, and lipids. Zooming all the way in, we see a single carbon atom, the very foundation of all life on this planet. This world is intangible. This world is so small that we can't even imagine it. So how, as a teacher, can we do this? I propose three things. We use hands-on tangible models. We use 3D modeling, digital modeling on the computer, and 3D printing to make this intangibly small world real. These three pictures here are examples of the models that my students are using. The first one on the left shows my student manipulating the atomic structure of a protein. Every atom on that protein structure he is modeling, and he understands how they work together. The middle picture is looking at the amino acids and manipulating the chemical properties, how each amino acid interacts with each other and the properties of each one. Finally, the students on the right are manipulating the three-dimensional structure of a protein. So this tiny molecule that we can't perceive is now in their hands in 3 dimensions. It's tactile. It's stimulating their senses. It's allowing them to become curious. It's allowing them to ask questions and be inquisitive. Beyond that, they can now model the 3-dimensional structure of proteins using an open source program called JMOL. Using simple text prompts, my students are able to manipulate the protein structure, to highlight different regions, and to change the way that protein is actually modeled. What that allows them to do is look at different aspects of the protein and highlight specific regions or interactions within that protein. These molecular machines are just that. They are machines, and every component must be understood if we are truly to understand the natural world at this level. The advantage of using digital 3D printing now is enormous. From this 3-dimensional model, we can make a 3D printed model. So now we have a real-world representation of protein. It is something where you can look at it, you can wonder, you can imagine what this is actually like. The challenge I gave my students was to link the macro world, the world that we all see with our senses, with that micro world. What I did is I challenged my students to identify the molecular basis of a debilitating disease known as cystic fibrosis. Cystic fibrosis is caused by a mutation in a single protein, the protein you see rotating on the left. There are over 1,800 different mutations that can cause cystic fibrosis. I challenged my students to select one and explain how that mutation in DNA can lead to the symptoms that we can all see of cystic fibrosis. This opportunity gave my students to become the scientists. They were the scientists. They were investigating this world. They were making the connections between the molecular world and the macro world around us. They understand cystic fibrosis not as a disease that has horrible side effects, but as a molecular mechanism. There's 1 or 2 atoms that are off, and that's the cause. This allowed my students to ask the questions, to wonder. One of my favorite quotes from this class, a student, a junior in high school, says, "Mr. Lane, I think I just found the edge of science." And he did. He had found the very edge of our understanding of where biology was. He was reading a peer-reviewed journal article and was making sense of it. He had found the very edge of science. Each class was allowed to print their own 3D model of their protein. They kept with them a 3-dimensional representation of their learning, of their understanding, of their investigation, their exploration. So what's next? Where do we go from here? What's next in biology is the idea that we could potentially make our own proteins. We can design our own DNA. We can engineer our own 3-dimensional protein structures. We can look at diseases not as symptoms, not treating the symptoms, but treating the molecular causes. We could revolutionize medicine. But in order to do that, we need to inspire a new generation of scientists and engineers. We need to let them think and be curious about the world around them. We need to remove the arbitrary constraints we place on our students and allow them to be the scientists, to wonder, to ask why, and most importantly, to say, I don't know. When they say those things, they open the door and they are allowed to wonder and investigate. So what's next? We are inspiring a new generation of students in the natural world. But the natural world is no longer just the tangible. The natural world includes everything from an atom to an ecosystem. Thank you.